The types of RNA. Okay, there was a three kind of RNA: mRNA, rRNA, and tRNA. Okay, what are the role? Why they are present? mRNA. Okay, mRNA always present. I told the DNA is converted in the form of mRNA. The double stranded DNA is going to convert to single stranded by involving of RNA polymerase inside the nucleus. That is a transcription. Once the mRNA single stranded transported through nucleophore, they will go on attached to the ribosomes. Okay, go on attached to the ribosome binding, that is called rRNA, that is ribosome RNA. So the transcription is processed over, the mRNA is coming out and they will go on bind with the ribosomes, that is called ribosomal RNA. Once the ribosomal RNA, they will make the amino acid sequence and protein formation. Okay, the protein formation, the amino acid, the attachment of amino acid and protein formation, they called as tRNA, that is transfer RNA. Messenger RNA, ribosomal RNA, transfer RNA. So messenger RNA to carrying all the message, all the genetic information from DNA to mRNA, that is carrying the information from nucleus to cytoplasm and transport ஆகுறாங்க. Then they will edit the formation of protein with the help of ribosome. The RNA is going inside the beat of ribosomes. ஸோ எப்படி ஒரு மணிக்குள்ளார பீட்ஸ் வச்சுருக்குமோ அது மாதிரி இந்த ரைபோசோம்ஸ் போயிட்டுன்னா ரைபோசோம்குள்ளே டிஎன்ஏ சிங்கிள் ஸ்டாண்டர்ட் போயிட்டு they will make amino acid sequence, then formation of protein, that is called transfer RNA. So மூணு RNA இருக்காங்க, that is for mRNA and tRNA. So mRNA is located in the formation inside nucleolus, that is transported into cytoplasm by the help of ribosome attachment. They will make amino acid, then amino acid sequence, they will code the proteins with the help of tRNA. So this is, then protein will be come out. This is the types of RNA. Okay, so that mRNA is single stranded. Okay, the single stranded RNA molecule carry the instruction from the gene, carrying the information from the DNA. Then it will go and bind. So, okay, the part of the structure of our ribosomes made for rRNA. Okay, this RNA going to bind. Okay, then rRNA is making for transfer to amino acid to make protein. Okay, then it will come formation of, again they form for the clever shape, the tRNA will form, make the proteins. Okay, these are the types. So you should know the difference between mRNA and tRNA. Okay, mRNA is for messenger carrying the genetic information. tRNA role is for transfer. Okay, so the mRNA is transcribed from DNA, make for ribosomes. The tRNA translated the mRNA to making new polypeptides and protein. Okay, it's a codon or complementary to DNA triplets. This is anticodon to make specific amino acids. Okay, இப்போ mRNA attachment ஆயிருக்கு ரைபோசோம்ஸில் வந்தவுடனே they will make codons இருக்கும், அதுக்கு ஒரு anticodons ஃபார்ம் பண்ணி they will make specific proteins, amino acids create பண்ணுவாங்க, that is anticodon. Codon getting from the mRNA and the tRNA, they will make anticodon, they will develop the amino acids. Okay, this is simple strand mRNA, that is single stranded. This is clever shaped loop structure. They will ask question, tRNA shape என்னன்னு கேட்பாங்க. The tRNA shape is clever shaped loop structure. Okay, அதே மாதிரி broke down after translation. ஏன்னா அது after translation ஆனிச்சுன்னா the mRNA will broke down ஆயிரும். But reactivation always, the tRNA, they will make reactivation, make amino acid after transcription. Okay, the gene is made up of triplet DNA base coding for specific amino acid. Okay, that is transcribed in the form of mRNA. The complementary base pair to convertible DNA to code the mRNA, make the polypeptide chain, and anticodons will be developed by tRNA, the codons from mRNA to make specific amino acid and proteins. இதுதான் நடக்கிற transcription translation process ஓட main important. The message getting from the DNA, the messenger RNA take the messages, then attachment to the ribosome, they will make tRNA, the tRNA make the specific amino acid with the help of codons from mRNA, Then the code will make anticodons that they develop for specific amino acid and proteins. This was the main difference between mRNA and tRNA. Okay. So finally, the holistic view, getting DNA, getting DNA to mRNA, mRNA to rRNA, rRNA to tRNA, then make the proteins. So this is the holistic process for central dogma of life. Okay, central dogma of life and structure of nucleic acid. Okay.